Hi, my name's Amelia, and welcome to my presentation, Testing the Fitness of Artemia franciscana in Certain Extremes. Today we'll be focusing on hypersaline environments, which is where brine shrimp thrive best. Our question for today is, how will changes in the Great Salt Lake affect future Artemia franciscana generations? Now, Artemia franciscana is a certain species in the Artemia genome. Artemia are all types of brine shrimp, while franciscana are the brine shrimp that actually live in the Great Salt Lake. So we're going to look at, first of all, my career competencies, which is a required thing for my personal project, which is what this is. So basically what I have to do is I have to figure out something that I want to do, like a career-wise, and then I have to figure out a project based on that. And then adding to that, I have to find 4-ish competencies, basically like skills that I've developed from this project, and show them. So I'm going to start with this quote real quick before I move on to my competencies. Brine shrimp have been present in the Great Salt Lake area for at least 600,000 years. And this is from the Utah Division of Wildlife Resources. So basically what this means is that from the Great Salt Lake's creation, which was ways longer than that, brine shrimp have been there for about 600,000 years. So basically what that means is that brine shrimp are quite an old species, at least Artemia franciscana is, and the past species that came before franciscana in the Great Salt Lake area. Now, my competencies, the 5 I picked are knowledge of core subjects, critical thinking / problem solving, digital literacy, career exploration and development, and financial literacy. You can skip past these, they really aren't that important to this video. Now, let's move on to the knowledge of core subjects. So basically, this is sort of like your science learning area. So what I ended up doing was I did a bunch of calculations. I calculated how much food these brine shrimp needed. I calculated the salinity from my salt type that I have at home. I did a bunch of calculations for the water chemistry to make sure that it was cycled properly and had the correct nutrients. I also ended up having to create a habitat, as you'll see later. It's quite barren, but that's normal for brine shrimp habitats, as they typically are used to floating in the water column and not staying in spaces, like well-developed and well-forested spaces. So as you can see, I have lots of notes. The middle one is salinity. To the right, you can see my setup on dates. I spent about 6 hours on that first day, just setting up everything. And then on the bottom, you can see a quick little description about how cycling works, where it starts off with ammonia, which is fish waste and excess food waste, and those break down into what's called ammonia as a toxic chemical, and it's actually very harmful to creatures in the environment, including isopods and other things like algae. It actually breaks down some of their silicate shells when we talk about diatoms later. And then moving on to the second stage in the cycle, we need bacteria to make this happen. So basically what bacteria do is they break down this ammonia into nutrients they can use, and then they leave nitrite as a byproduct. Nitrite is actually more toxic than ammonia, and it's because it's a byproduct of bacterial activity, means also we need a bacteria to get rid of the nitrite. So nitrite becomes nitrate after bacteria take what they can from it, and nitrate is actually used for aquatic plants and other plants in general. You know, if you look at fertilizers, you'll see that they have a large nitrogen compound, which is actually what helps them let plants grow. So nitrates are mostly harmless. You can go up to around 80 parts per million, I do believe. You can correct me if I'm wrong. But on the bottom to the left, you can see that my ammonia was about 2 parts per million. To get that to 2 parts per million, I needed 4 drops per gallon. And this is a fishless cycle, which basically means that I'm not putting any animals in until the tank is fully cycled. And that took about 48 drops to get the ammonia to 2 parts per million. And then I ended up having to critical think and problem solve a bunch for this project. I solved more issues than I was expecting to, including cycling problems. There was a mass die-off at one point, and there was a non-viable batch. And we'll go over all of these and more in my problems and solutions section. So basically what ended up happening was that I decided on solutions through my study and just through what I had that could work. Now, moving on to my digital literacy, I used mostly Google Scholar for this. I chose the most reliable sources from Google Scholar, and then I actually also uploaded to YouTube for the first time, which is this video. So there's a big empty space, but that's where this thumbnail will be. Career exploration and development, pretty much just discovering marine biology. I've always loved marine biology and I just want to learn more about it, and I thought that this project that is required for me to graduate is just a great segue into, you know, the career I want to become a part of. So what I ended up doing was I developed a bunch of career-related strengths throughout my project, which I'll discuss later, and then I learned about the biomes and habitats in which species like brine shrimp live. And then financial literacy. I ended up having to manage a bit of a budget. I tried to stay within about a $750 price range, although the tank was most of that cost, just because it made a big dent in, glass is very expensive. I ended up choosing between price and quality more than I like to admit, unfortunately, just because a lot of the good quality things are way more expensive. So I ended up going with what I could find for a good quality for a good price. Moving on to career, I also have to demonstrate what career I'm researching and how it goes to my project, basically. And before we do that, I'll read the quote real quick. Scientists believe that they arrived as cysts on the feet and within the feathers of migrating birds. Basically, this is talking about how scientists believe that brine shrimp arrives as cysts, which we'll talk about later. And they actually ended up getting stuck in the feet and feathers of migrating birds, and migrating birds are actually a very important part of this whole system, so we'll get into those later as well. So marine biology, the average salary is about $80,000, and that's based on experience. You know, if you're less experienced, you will get probably less pay. If you're more experienced, you'll get more than that. Your base education is a bachelor's degree, but it's pretty much normal to get a doctoral degree. It's typically expected at higher level positions. And there are hundreds of possibilities for careers in marine biology. It's really just based up to what you want to do within said career. Like, for example, I'm looking more into genetics in marine biology, which isn't the largest career, but I think it would be interesting to study. And then moving on, it's actually likely to grow by 5% by 2034, which isn't the largest increase, but it is an increase overall, and that's good for the career itself. And then our AI outlook for this career is that AI is actually a useful tool for science, and it really doesn't damage any jobs in the science area just because we're learning along with the machines, and AI actually relies on human input, which means that it can't really take over because it only knows what we feed it, and we only know what we know. And a few examples for what this could be used for is fish identification and trash identification. So basically what we can do here is we can compare trash to fish. So if we send out a bunch of ROVs, which are basically water rovers, to collect trash, they can identify what's a trash and what's a fish. And that bottom picture is an image of an AI-piloted ROV. I'm also very interested in studying this career. You know, I grew up loving the ocean. I always have. I really like anatomy, and biology is one of my favorite subjects. So adding this all together, it really just shows that marine biology is a great course for me. Moving on to a bit of a background on this project. You can see on the right, there are several images of the lake over time. So in 1986 is when it reached its record high of 4,211.65 feet, and in 2021 you can see it was its new record low, which was a 4,191.3 feet, and then in 2000 it's about average at 4,202.2 feet. You don't really need to remember those exact numbers, just remember how much of a difference there are between them, and even then that's considered a great increase and decrease between them. So I'm going to go ahead and read this quote real quick. As salinity increases and oxygen tension diminishes, hemoglobin synthesis increases. This is from an NIH article that I found on how Artemia are adapted to survive hostile environments, unlike any other species really, besides a few other microorganisms. And basically what this is saying is that brine shrimp can actually survive a bit of an oxygen depletion because they are able to make more hemoglobin than other species, and hemoglobin is the stuff that makes is the stuff that your red blood cells create, so they actually have more red blood cells than the average species. And this hemoglobin actually collects and holds on to oxygen, which allows the body to use it for longer periods. So we're going to start with an interview I had to conduct. So I ended up actually having to conduct an interview for my project, and what that looked like was talking with someone from the Great Salt Lake area. Her name was Grace Romney. She's a researcher at Grow the Flow, which is a nonprofit looking to save the Great Salt Lake from its drying out. And what we ended up talking about was a lot of interesting things. We didn't really talk about brine shrimp, but I was more interested in the Great Salt Lake at this point because I didn't have as much information as I did on brine shrimp. So what I talked about mostly was was harmful impacts from drying lake bed, like the toxic particles, and we'll go over those in a little bit. And then also we talked about other examples of hypersaline lakes that have dried up in the past. And we just overall talked about all the problems that the Great Salt Lake is going through, and it was a really invigorating experience. And I would very much suggest that if you're interested in this, go ahead and check out their website, help them volunteer. It's really a great program, and I would totally recommend it if you're in the area. Now, moving on to the Great Salt Lake itself. Great Salt Lake, as it is aptly named, is a hypersaline lake. It's one of the largest stops in the Pacific Flyway, which we'll go over in a bit, but basically it's one of the largest migratory bird paths in North America. And this lake provides irrigation for the city. Well, not the lake exactly. The rivers that feed the lake provide irrigation for the city. So that's part of where our issue comes in, is because the city is actually taking too much water for irrigation and housing, that the lake is actually drying up. It's also the largest lake in the Western Hemisphere, and also, of course, the largest hypersaline lake in the Western Hemisphere. And you can see on the top right, there's a water level chart. This was a screenshot on October 13th, and it was from the Grow the Flow. It's a great website, and even if you're not in the area, it's cool to check out their lake tracker because this is on the top. As you can see, on October 13th, it was about 33.5% of its maximum, which was about 4,202, which is a lot lower, unfortunately. And as you can see on the bottom left, you can see how elevation has changed throughout the years. That blue zone is healthy, so there's no environmental impacts, there's no danger really, there's no issues if you play in this lake. The transition area is kind of in the middle. It's mostly healthy, but in some areas it might be a little dangerous. And then the adverse effects is definitely where you're getting into the toxic area, like the dry lake beds becoming visible and the winds blowing up some particles. And then the serious adverse effects is pretty much when the lake is getting drier and drier, and it's getting almost too dry, and these particles are becoming more and more common and just more and more deadly as they keep growing. On the bottom right, you can see there's another graph, and it shows from October 13th the current lake level, and it shows that really the only thing that is doable at this time is hunting. The rest either has no data or it's very unsafe. So you can see for brine shrimp harvest in the north and south arm, it's very bad, very, very not optimal. And then in the biology section, you can see brine shrimp actually has no data, just because they're not actually really studied in the wild. They're more used for science research projects. Now, moving on to Owens Lake. Owens Lake was a saline lake in LA, and this is actually the lake that my interview person actually guided me to, and I don't think I would have found it without them, so I thank them a lot. This lake dried up in 1926 due to river diversion for irrigation and housing, very similar to the Great Salt Lake. However, this lake is a lot smaller. This lake was only about 110 square miles at maximum fillage, and because of the river diversion, it just slowly drained until it was completely drained in 1926. It's the largest carcinogenic particulate dispenser in North America, which basically just means that it releases the most toxins considered carcinogenics in the entire area of North America. And this lake bed is actually 100 times the federal pollution standard, which means it's 100 times over what the U.S. considers a pollution. Now, continuing on Owens Lake, they've actually tried to refill it several times, and refilling has cost over $2 billion, so occasionally they fill it up again. However, without the two rivers feeding it constantly, it just keeps drying out due to evaporation and just water penetration through the soil over time. And unfortunately, this method is really not sustainable. With the water requirements required, it just really, you can't expect this to happen in the near future, especially since recent droughts have been hitting the area for several years now. And also, they've had to do 45 miles of dust mitigation around the lake, which means they cover the dry lake bed in rocks, they plant grasses, they cover it in sand, really anything to just mitigate that dust problem that brings up toxic chemicals. And you might be wondering what that red stuff is. We're going to go over that in a bit on the brine shrimp section. But that's actually halophilic algae, which means that it's salt-loving algae. And that's typically the only thing that grows in those sections, because it's saying that this is super salty and not really anything else will grow there besides a few bacteria species and that red halophilic algae. Now, our final example of a dried up hypersaline lake is the Aral Sea, which actually isn't a lake, it's a sea. It was between Kazakhstan and Uzbekistan when the Soviet Union diverted the main rivers to irrigate cotton fields nearby, which actually caused the slow diminish of the lake, as you can see on the top right. As you can see, the South Aral is more saline, so if you look to the left on that 2020 picture, you can see that there's a little bit of water all by itself. And that is where the south is located, and this actually is more saline because there's no direct water flow, whereas the north has a bit of a river left, so it gets a little bit of fresh water every once in a while. Now, as you can see, a lot of the sea is gone now, so the toxic dust is exposed on the seabed. So what happens is that natural deposits of minerals like mercury, lead, cadmium, they naturally occur in seabeds because the sea, it starts to erode at the lake bed over time, and that leaches the chemicals that are in the ground up into the water, and those eventually settle on the lake bed. However, many people, typically in the older times, would dump toxic chemicals, toxic waste into these lakes and seas, and that actually increases the amount of toxic stuff in the lake bed. And only about 10% of this Aral Sea, 10% of the Aral Sea remains, which is a great reduction from 1977 before it was touched. And on the bottom you can see ships that were unfortunately lost due to the water reduction. Now let's move on to Artemia, which are brine shrimp. I'm going to go ahead and read this quote real quick. Artemia can be considered a model animal extremophile. Now extremophile means extreme loving, and in this case they're talking about how they love the extreme salinity. And this is also by the NIH article. First, let's look at a phylogenetic tree. So as you can see on the top, there are codes, because this website that I used uses codes to label the proteins that you're looking at on certain species. So let's go ahead and focus on franciscana and salina for a bit, which are the only two that don't have a 9CRUS at the end. And as you can see, they're at the very top, and you can see that they split off first compared to the other three, or those are quite close relatively, which shows how these two species ended up actually splitting very, very late, or very, very early in the past. Now, Artemia salina actually lives in the Mediterranean, while Artemia franciscana lives in this Great Salt Lake. And it's quite interesting that these two locations, despite being so far apart, have two more similar species to each other than to either of the others. And then as you can see, of course, the bottom three, we don't really need to focus on those ones, but those are just the other species in the Artemia genus, and you can see that they're a lot closer than the others. Now, let's look at the identity matrix real quick, and again, we're going to focus on franciscana and salina. It's just interesting to include the other 3 species. And as you can see, if you look to the very right in the Artemia franciscana, you can see it's compared to salina, and they have a 96.28% identity, which means that these 2 are actually really close together compared to the others. You can see also that Artemia armenia and Artemia tibetiana are actually also very close with a 97.61%, but we're going to focus on the salina and franciscana. Now, these 2 are very close, like I said, because they split off so early, and also because they've been put in their own environment that's perfect for them, whereas the others have had changing climates and areas over the years. Now, some basic knowledge on Artemia franciscana. Economic use. These brine shrimp are actually used for things like aquariums. You feed your fish typically with these. They actually feed a bunch of hatcheries. They're just really beneficial for the economy in the Great Salt Lake area. And they're a food source for migratory birds, which we'll go over at the very end. But basically, these birds use the brine shrimp to fatten up for their long journey to the south. And Artemia franciscana is the native species to the Great Salt Lake, and they're the largest inhabitant, which means they're a macroscopic organism. And macroscopic just means that you can see them with the naked eye. They're not minuscule. Now, you can see on the bottom, I have these places labeled, and there's a diagram of brine shrimp. The bottom is 2 images of the Great Salt Lake. You can see that middle divider on the left, and that's from the old mining days when they were using efficiency to get as much ore as they could to sell. So they built this road that unfortunately separated the 2 sides and caused the north arm, which is where there are no brine shrimp, to get very, very red. And as you know, this is from the halophilic algae, which are pretty much the only thing that can live there besides a few other microorganisms like bacteria. Whereas the blue side is where the brine shrimp are actually found, and this is because it's a better salinity quality. There's fresh water moving through it, through a small river, which allows the salinity to stay at an okay salinity. And then the franciscana's adaptations are advanced osmoregulation, which means that they can take out salt 10 times saltier than seawater. So basically what osmoregulation is they separate salt from the water so that they can expel the salt and use the water to hydrate themselves. They are also extremophiles, which we went over earlier. It just means they have a love of the extreme. In this case, they're halophiles, which means they love salt. They can choose between oviparity and ovoviviparity, and oviparity is where gastrulated encysted embryos come in, which are commonly called cysts. And to make cysts and to keep them, it takes about 324 protein-coding genes in the brine shrimp's DNA, which is a lot of genes. In ovoviviparity is live birth, and as you can see on the top, those oranger eggs are actually a live birth, while the darker ones on the bottom are encysted birth. And then to the left of the encysted female, you can see the male who has claspers on his head. These mature brine shrimp actually reproduce about every 5 to 6 days, which means that they're really trying to increase their population before the birds eat them each year. Now, cysts are encysted gastrulated embryos, which are just commonly known as cysts. They can survive anhydrobiosis, which just means that they can survive outside of the water for an indefinite amount of time, pretty much, as long as the temperatures are not too extreme. So you can see on the bottom there's a little bit of a diagram where there's a cyst. Once you hydrate it, or once it gets hydrated, it actually needs light to hatch. And what you see from that is that the cyst starts breaking once there's light, and you can see the embryo slowly forming and turning into a nauple, which is basically the post-embryonic stage of brine shrimp before juvenile stage. And these cysts are considered a gene bank of past population conditions, which basically just means that they reserve they basically store the information of the past generations before this brine shrimp, what conditions they should take in to hatch and which ones they should pick. And then females perceive conditions and choose whether to have live birth or assisted birth. So basically what happens is they use their bodies have these antennae, and they use those not only to sense water movement, but also to sense the chemistry of the water and determine whether or not to have live birth or assisted birth. Now, cysts can form from both sexual reproduction and asexual. However, they are the only thing that can come from asexual reproduction, as live birth can only come from sexual. Some interesting facts about brine shrimp. Brine shrimp can actually benefit from cestode parasitism. It actually increases their resistance to arsenic, which is a bit odd because cestodes, as you can see in the image, are actually tapeworms, and it's a bit odd that tapeworms, when infected with tapeworms, a brine shrimp can survive more arsenic, but you do you. And then secondly, Artemia are used as a water quality tester in several areas of the world. So basically what they do is they hatch these brine shrimp, they let them into the water, and they see if they survive or not. And that's basically just a test to see if there are any toxins in the water, to see if the water is just relatively safe, because Artemia are actually highly sensitive to really just any changes in the water. And typically they're the only macroscopic organism, which we already talked about, macro meaning large and scopic meaning visible. So basically they're just saying it's the only visible species besides like algae and bacteria, which you can't really see bacteria. So, and then the nauplii, which are the post-embryonic stage of brine shrimp, they're very sensitive to mycotoxins, which are toxins released from fungi. And these are very detrimental to humans and other animals alike, so it's really interesting that we can use these as a research to test what's going on. Now finally, we're going to talk, not finally, but now we're going to talk about my experiment, and I'll go ahead and read the quote real quick. In a few cases, Artemia is the only macroscopic representative. This is also by the NIH article, and that's basically saying what we just talked about, how Artemia is really just the only species you can see with the naked eye. And to be fair, they're quite small. They're only about 2 inches once they get to full maturity. Now, moving on to my original procedure, my initial plan was to cycle the tank, which is in the top, and then I was planning to hatch the shrimp, and then I would slowly add salt once the first few generations matured, and then I would observe their new behavior to see what would happen. Now, unfortunately, I had a bit of a cycling problem. As you can see, the middle images, I just kept getting those results over and over again, so I stopped taking pictures of them. The top left, is what my tank originally looked like after I dosed the ammonia. You can see it's about 2 parts per million, and the other 2 are at 0, and then it just kept looking like that for a long time, and then finally I figured out what was going on, and I brought it back down to 0 on all fronts. So before that, though, I decided to add live media, which are just clay ceramic rings that have bacteria living in them, and I got a few hitchhikers. Thankfully, they weren't any detrimental parasites or anything. I just caught a bristleworm, a very, very small juvenile bristleworm that unfortunately died, and then a few scuds that also died because I put them in not dechlorinated water, and that was a very, very big problem on my part. And then moving on to hatching the shrimp themselves, you can see that I ended up hatching them in this tall cylindrical hatchery, and right now they're about in between the final nauplii stage and getting into the juvenile stage. So they have more than 2 legs at this moment, but they're still very small and they definitely cannot reproduce. Moving on to some problems and solutions I had, I'll go ahead and read the quote real quick. The brine shrimp fishing industry brings in $70 to $100 million annually. This is actually, unfortunately, an older statistic from Learn.Genetics, so I have a newer statistic later in the presentation. Unfortunately, I faced some cycling issues when the tank wouldn't cycle, and occurred for several weeks, and it turns out I actually overdosed the bacteria. I put too much ammonia in to start with, and that kind of hindered the system. So what I did originally was I added live media, not knowing what my problem was, and then I finally realized what my actual problem was, so I fasted the bacteria for a week, which just means that I didn't add any more ammonia. I did a quick little water change, and then it cycled within days after, as you can see from that top right image. And then unfortunately, I also had a non-viable batch. I let this batch sit for about 4 days, but unfortunately there were no signs of life. As you can see from that bag on the bottom, there was mostly salt. There weren't very many cysts, and so what I did was I ordered a new batch, and the new ones hatched within 2 days. My guess on what happened was that they got cooked in shipping, and they really just couldn't hatch because of that temperature flux. And then unfortunately, after all of this, there was a mass die-off, and it turns out I ended up overfeeding them, and there wasn't enough dissolved oxygen for them all to survive. So what I ended up doing was I added another air pump, and I siphoned out the dead and extra food. And then what I also ended up doing was I fed less per feed, and I ended up stopping feeding them because of my next slide, which is an algal bloom. And these brine shrimp have actually been consuming this algal bloom after I scraped off all the dead parts. So what ended up happening was that this algal bloom caused an anaerobic pocket near the bottom, and Artemia actually stay mostly near the bottom, at least mine did, and it caused a bunch of suffocation problems, and a lot of them died from that, as well as the overfeeding issue. So what I did was I siphoned out the excess algae and scraped most of it off. I left a little bit of the living stuff so that they could eat it. And then unfortunately, in my 6-week period, I was truly unable to finish my project. Unfortunately, most of my brine shrimp died. Cycling took way too long. The brine shrimp actually take weeks to mature. So I pivoted to more of a research-based project. As you can see on the front, or the closest left-right image, that big brine shrimp is actually magnified, and that's what they looked like 2 days ago. And then on the top right, you can see some embryos are coming out of the cysts, and then on the bottom, you can see the unfortunate algal bloom. Now let's go ahead and move on to impact of environmental changes. I'll go ahead and read the quote real quick. In 2022, Great Salt Lake levels broke record lows, and that was when it hit about 4,108 feet, which was incredibly low and very unexpected. Well, I wouldn't say very unexpected, but just not really good for the environment. That's from USGS, which you can find on my works cited page at the very end. So we're going to start off with Owens Lake. Owens Lake was 110 square miles at max, and it caused toxic dust clouds when it was completely drained, and which happened in 1926. And that bottom image is a picture of the dry lake bed in the Great Salt Lake. Now, going ahead and looking at the Great Salt Lake is actually 3,300 square miles at max, which is about 30 times the size of Owens Lake. And unfortunately, it's already spreading toxic particles throughout the air, even with not that much missing. And only 33.7% of the lake bed is covered. Now, when we look at the height of the lake, it's taking it from the lowest point, so when you see that it drops about 20 feet, you're actually losing a lot more space than what it's saying, just because it's taking from the lowest point, and there's a lot of shallower parts than the lowest point. So basically what's actually happening is we're losing a lot of area that's not really being shown very well to the general public, and I feel like that's why most people really don't take this as seriously as they should. Let's go ahead and look at some restoration cost analysis. So looking at Owens Lake, you can see that it was around $2 billion for 110 square miles, and that was just to refill the lake. And they had to use about 45 miles for dust mitigation, which we talked about is planting plants covering the dry lake bed in rocks, really just trying to stop that dust from getting into the air. And then finally, let's go ahead and look at the Great Salt Lake's cost analysis. It's estimated, of course it's an estimation because we can't exactly know for sure because we haven't even tried to save the Great Salt Lake. Now, it's an estimated $1.5 billion in dust mitigation alone, and that will actually have to be around $15 million annually after that initial cost just to maintain this mitigation practice. And then using Owens Lake as a basis for around $2 billion for 110 square miles of water restoration, it's going to be about $80 billion to restore the Great Salt Lake to what it once was. Now, let's go ahead and look at the harm that can come to the brine shrimp themselves. Because of an inbound salt-to-water ratio, it just means that there's more salt per density of water, which just makes the water saltier, and they really they can only handle a certain amount of water, which is really shown if I could finish my project. Unfortunately, I couldn't. And then let's look at the salt increases from 50 years ago to today. So the north arm, which is the one that's blocked off from the fresh water source, actually increased by about 25% in the last 50 years, and the south arm increased by about 12% in the last 50 years. So my prediction is that the south will eventually be uninhabitable just because of this increase over time. And then, now I told you I was going to talk about bird harm and bird migration. So you can see that circle in that poster. That's about where the Salt Lake is, and about 10 to 15 million shorebirds pass through the Great Salt Lake on their trip to the south. And what happens there is it's one of the largest stops in the migration pattern called Pacific Flyway, and these birds consume brine shrimp and brine flies, which are just flies that like salt water, and they try to fatten up as much as they can on them, because this is the last stop before a nutrient deficit, which basically just means that this is the last stop before you lose a lot of the nutrients you can gain. There's no more really big stops. You can't really get much food anymore after this until you finally make it to where you're headed, which is really unfortunate for these birds because it means that a lot of them will have to pivot to different migration patterns, and really it'll just affect the whole ecosystem. And then let's look at the Salt Lake City harm. So this city can actually face a lot of harm from this as well, not only because these toxic particles will affect the city, but also just because of the industry harm. So that old statistic said that about 70 to 100 million, I do believe. Unfortunately, now it's closer to 60 to 67 million per year in the industry, and about 90% of these brine shrimp that are raised are sent to Asia for commercial use in basically just hatcheries and fisheries, which profit from shellfish, fish, mollusks, you name it pretty much. Anything that you can eat, they probably hatch there. They're also used in scientific research. They test them for toxicity testing, like we talked about earlier, and they're used in developmental science and genetics. And if you're wondering why I put that brine shrimp in an astronaut photo there, or an astronaut suit there, it's actually because brine shrimp did go to space once on a NASA project just to see how they would survive and exist with their extremophile tendencies. Let's go ahead and look at some chemical toxins released from the Great Salt Lake. So basically what we're seeing is that lead, cadmium, arsenic, and mercury are the main attackers. The PB is lead, CD is cadmium, AS is arsenic, and HG is mercury, if you're wondering what they look like. These particles become airborne from wind and just air in general, and these particles can actually cause birth defects and cancers, and one of the largest issues from these is pulmonary distress, and this specific pulmonary distress is called chemical pneumonitis, and these particles can travel over 100 miles a day. And then that bottom image is about how this lung distress can affect your breathing. You can see the alveoli becomes enlarged, and that affects how you take in oxygen, I do believe. And then more on toxic particulates. The Salt Lake City area suffers from smog, as we can see in that top right corner. Photochemical and inversion smog types affect the city, and we're gonna go ahead and start with photochemical. And photochemical smog is when sunlight reacts to pollutants such as vehicle smoke, vehicle exhaust emissions, wood stoves, industry emissions, and wildfires. And then the sunlight actually changes these through chemical reactions through the heat of the sun, and it really just makes these worse, and it turns it into that blanket of dense smog that we know today. And then inversion smog is when the temperature traps particles, and this typically happens in the winter because what happens is that warmer air or colder air gets stuck underneath warmer air, and it creates this thick barrier that the particles cannot travel through, and it makes this dense layer of smog. And this smog, unfortunately, will worsen particulate damage from the Great Salt Lake just because this dust, as you can see on that bottom poster, it releases salts, toxic metals, and alkaline particulates that get stuck in this smog. And because in inversion smog, which typically happens in the winter for the Great Salt Lake area, unfortunately the cold is much better at holding in smog in a certain area, which also means that it'll hold in these toxic particulates a lot better, which is awful for this area. And then in conclusion, building survival ecosystems is difficult, as shown by my tried experiment. I really just couldn't get it started. I really just couldn't get it finished in the 6 weeks time that I had. Unfortunately, the Great Salt Lake will soon be uninhabitable unless we do something about it. And my prediction is that toxic clouds will soon poison the city, causing a bunch of respiratory diseases, cardiovascular problems, lots of just cancers in general, and it's really just going to harm these people. And unfortunately, I'm going to predict also that the city will face economic loss just due to that brine shrimp industry falling apart. And then finally, my guess is that organism populations will decline because unfortunately, just like the toxic clouds that will cover the city, these toxic particles can travel over 100 miles a day, which just means that they're going to spread everywhere and to everyone. And unfortunately, this will harm a lot of more species than we're expecting it to. Finally, these are my works cited. Feel free to pause whenever. I'm just going to go through these real quick. I really enjoyed doing this project, and I think that it was a great way to learn about just brine shrimp and the ecosystem, how it's declining in general. So thank you very much for listening, and I hope you enjoyed this as much as I did.